Hey guys, this is Kendall Terry, and this is a look at DNA, the genetic material, and this is the replication edition. So what is DNA replication? Well, since Watson and Crick gave us the shape of DNA as a double helix in 1953, they proposed that one of the strands would serve as a template for a new strand. So they proposed that the DNA would separate and that one strand, the strand that was there, would serve as a template for a new strand. Shortly after this, it was proven that they were correct, that every cell in your body has the same DNA, and that all came from an original cell, which was the egg cell after it was fertilized by the sperm. And as that cell then began to divide, it had to pass the DNA in that cell down to every cell made after that to make up all the complex cells in your body. So how does it do that? It gets the same DNA by separating the DNA. You can see that here in the picture. Separating the DNA and using each strand from the original as a template to build the new strand. So each strand then from the original one is a template for the new strand, and then they just simply bind using base pairing rules. If there's an A on the original strand, then the new strand has to have a T. If there's a C on the original strand, the new strand has to have a G. They have to match up the same way that they did in the original strand. And this method is called the semi-conservative method. You see that right here at the bottom, semi-conservative method, where the two strands will separate and then each one will serve as a template for the new strand to be made. So how does that take place? Well, there's three simple steps in this. The first thing is the DNA helicase. If you'll look here on your screen, you'll see kind of like this purplish colored bubble on here. That represents an enzyme, and that enzyme is DNA helicase. DNA helicase is responsible for unwinding the DNA. Remember, it's in a double helix, so it has to be unwound, and when it unwinds, it will separate the DNA from each other, and that separation is breaking those hydrogen bonds between the nitrogen bases. And you see the nitrogen bases sticking off right here, and it's because they've been separated. Those hydrogen bonds have been broken by DNA helicase so that it creates a gap. Okay, now there's some other proteins that aren't important to our to biology 1 right now that will keep them separated and won't let them go back together. And while they're exposed, the next step will take place. This creates what we call a replication fork. And you can think of that as kind of an upside down Y, but there's the DNA here, it's being separated and unzipped, and that creates a fork in the DNA and it exposes these nitrogen bases right here. So another enzyme called DNA polymerase, you can see that right here, DNA polymerase. In the picture, it's kind of like this greenish, bluish bubbles. DNA polymerase will move along the DNA strand and add nucleotides to the exposed nitrogen bases. And it simply follows what we call base pairing rules. These were the same rules that Watson and Crick used to build DNA. They got this rule primarily from the work of Erwin Chargaff. We've discussed that in a previous lecture. So he said that A is always in equal amounts of T, so A has to be bound to T. C is always in equal amounts to G, so C has to bind to G. And this creates two new double helixes. You can see those right here. The third step is DNA polymerase will be able to go back and proofread the DNA. So DNA polymerase will proofread the DNA, and it will check for any kind of error in the chain. And if it sees an error, it will take that error and fix it. And because of this, DNA polymerase has an error rate of only about 1 in 1 billion nucleotides that it adds. So the error rate is very, very, very minuscule or small. And that's good, because otherwise you would end up with a bunch of mutations. A mutation is simply the wrong DNA sequence. So if you've got a change in DNA, that could be very bad or detrimental to your health and could lead to the onset of stuff like cancer. So DNA polymerase is able to go back and check and make sure there's no error, and if there finds an error, fixing that error so that it's no longer there. This gives you two new identical, and I can highlight identical here, identical DNA molecules, each made of one new strand connected to an old strand. You can see that here in the picture. The old strand is the blue strand that comes down here. The new strand is this green strand right here. And both of these are identical to each other. That's at least the goal, because later on when we talk about mitosis, these will be separated and put in different cells. So each cell will have an identical strand of DNA.